join us, hopefully, soon. And we'll start with the first presentation by Saad Al-Harbi, titled Saudi Ophthalmology Residency Training, Evaluation of Resident Satisfaction and Comparison with International Standards. Saad.
Assalamu alaikum. I'm here today to present a study, or the result of study, that was done to evaluate the satisfaction and effectiveness of Saudi ophthalmology training program in preparing residents for entering clinical practice. And to do that, all graduates from Riyadh, Jeddah, and Dammam in 2019 and 11 were invited to participate in a 43-item survey designed by Zuhu and his associate with reference to the competencies outlined by the International Council of Ophthalmology for Training Program. And today, and for the sake of the time, I will just concentrate on the clinical and surgical training with quick overview and touch on research. In 2009 and 10 and 11, 58 residents graduated from Saudi Ophthalmology Training Program. Half of them were from Riyadh Center. 50 out of 58 residents were participated in this study with a response rate of 86%. 20% were female and 3 only were non-Saudi. 2 of the graduates passed the board exam and 1/3 failed to do that. Generally, most residents felt that the amount of time allocated to clinical training during residency was adequate. And most of them were satisfied. Clinical-based training across most subspecialty was generally considered satisfactory. However, a significant proportion of residents stated that they had insufficient exposure to areas of oculoplasty, neuroophthalmology, refractive, uveitis, and low vision rehabilitation. Indeed, these areas were identified as areas requiring further training. 44% of residents stated that the amount of surgical-based training is little or even too little, and this was reflected on their satisfaction since only 1/4 of them were satisfied. Exposure to the surgical aspect of various subspecialty was insufficient in most of the time, and the answer was expected when we asked them about their ability to comfortably done this list of procedure, and most of them were able to do only ECCE and pterygium. In order for residents to achieve comfort, competency, safety in performing various procedures, it's important for training programs to ensure that residents did adequate surgical volumes. And as we know, cataract is the leading cause of reversible blindness in our country and worldwide. And it was shown that the complication rate were significantly reduced after residents had completed 80 cases or more of phacoemulsification. But unfortunately, only 1 resident did more than this number, and almost half did less than 30 cases, and some did even less than 5 cases. When we joined phaco with ECCE and we label it as a cataract, and we compare it with the Canadian example, we found that most Saudi residents did less than 100 cases, while only 2 Canadian residents did less than 100 cases, and 50% of them exceed 300 number. For trabeculectomy, Riyadh's resident did a comparable number of cases to Canadian residents, but 1/4 of Jeddah residents and 1/3 of the MAMS residents finished their residency without doing a single case of trabeculectomy. And in the field of strabismus, more than 90% of Saudi residents did less than 20 cases, while 2/3 of Canadian residents exceed 30 cases and even reached up to 60 cases. In general, about half of the Saudi residents were unsatisfied about their operative experience in terms of number, complexity, and variety. And when we asked them about their ability to do some aspects of competency that was requested by the International Council of Ophthalmology for the training program, we found that only 1/4 of them feel comfortable to prescribe glasses, 1/3 can do phaco comfortably, 2/3 can do ECCE, and only 6% can lead simple lid procedure like ectropion, intropion. 6% only can do simple lacrimal surgery like DCR. Fundus exam can be done by all, but detailed fundus exam with scleral indentation can be done only by 60% of them. And unfortunately, only 18% can screen for retinopathy of prematurity. 1/3 can manage in regarding of treating and diagnosing uveitis, but most of them can do YAG-BI and pan-retinal photocoagulation. In touch for research, on the research part, 56% of the residents did at least one research, but half of them finished their residency program without even doing a single research or writing a single paper. And generally, the satisfaction rate among Riyadh residents was 2/3 of them, but 16.7% of the Jeddah residents and 23% of the MAMS residents were satisfied. The other were either semi-satisfied or 1/3 of Jeddah and the MAMS residents were dissatisfied. Doubling the number of residents from 11 in 2009 to 20 in 2011 associated clearly with dramatic drop of satisfaction from 80% in 2009 and further to 30% in 2011. And this was reflected obviously on the Saudi board exam failure rate, which increased from 10% in 2009 to 60% in 2011. In conclusion, Saudi ophthalmology training programs show a high rate of discrepancy from center to the other, with generally unacceptable satisfaction rate in Jeddah and the MAM centers. And yes, we are in need for more ophthalmologists to prevent blindness in our country. But before considering increasing number of residents or opening new centers for training, a clear strategy should be implemented to ensure a good quality of the outcome. Thank you.
Thank you very much, Dr. Saad. This is a very interesting presentation, actually, and I'm sure it will or it will initiate a lot of discussion, hopefully, if we have time. We're running quite late. We're sorry for the delay. It was out of our hand. As we've seen, the opening ceremony was delayed and took longer than expected. So we will try to go through the program as quick as we can, and I would really urge the presenters to keep the time, and if they can even go shorter than the time allotted for them, we'll be really grateful as we have another session after the break. The second presentation is by Dr. Munder Al-Alwan. It's on stem cell research at King Faisal Specialist Hospital and Research Center. Dr. Munder.
First, I would like to thank the organizer committee for giving us the chance to present the stem cell research from King Faisal Specialist Hospital and Research Center. And secondly, because I'm the first presenter about this in this session about stem cell, so I'm going to go a little bit basic about stem cells for those who are not familiar with it. And I'm really, really happy that you bring this topic to the stem cell research and its application in eye disease. So a stem cells has two cardinal features: their ability to replicate itself, or what's known as self-renewal ability, and their ability to differentiate into specialized cells, neurons, muscles, or other cells. But what is the source, the common source of stem cells? There are two common sources. Embryonic stem cells, and these are controversial source of stem cells in most country and most religion, which is coming from embryo or fetal tissue. Basically, they come from fertilized egg. They take the inner mass of the blastocyte, and then they differentiate these into a different tissue. But there are other non-controversial stem cells, which basically coming from, also known as adult stem cells, and they're coming from rich source such as umbilical cord blood, bone marrow, and peripheral blood. And more recently, there was another source known as induced pluripotent stem cells. And the significance of this is that you can bring adult stem cells into an embryonic stem cell features without going through the ethical issue. And the significance of this has been awarded a Nobel Prize for Shinya Yamanaka, and 2 years ago has been awarded King Faisal International Award for his achievement. Basically, the induced pluripotent stem cells, what you do is basically all you need to do is to take a skin biopsy. You can differentiate these, or you can get the fibroblast of these. You can induce 4 transcriptional factors listed here into these cells, and you can convert them into induced pluripotent. And the beauty of this, these are patient-specific, so you can differentiate them into whatever you want under certain condition. You can inject them into animals to study their physiological relevance, resistant to certain drugs. You can also do in vitro drug screening on them in vitro, and whatever resistant you can inject into the patient, or you can do gene therapy on them and transfer them back to the patient. So is stem cell therapy is feasible? And the answer is yes. There are a lot of successful examples of using stem cell therapy in malignancies such as leukemia, lymphoma, in immunodeficiency disease such as severe combined immunodeficiency disease, inherited hematological disorder such as thalassemia. But because this is an ophthalmology meeting, so I thought to put this slide for you. Also, there are a lot that has been done on stem cells and eye disease. I'm not going to talk in details because the next speaker, Professor Ting, he will talk about this. But there are some successful results, really. What they do is they differentiate, induce pluripotent stem cells into retinal cells. They induce embryonic stem cells also to an optic cup, and they could also, in a clinical trial, they managed to use embryonic stem cells for degenerative eye disease, and the result is very promising. But the hope is to be able to use these stem cells for an irreversible disease, or disease that has an irreversible tissue damage, such as neurodegenerative disorder, spinal cord injury, heart disease, and of course, eye disease. Alternatively, also you would be able to use these stem cells to make a whole organ, which will give a chance for those waiting for organ transplant. This is a busy slide. I don't want you to remember anything, but this is just showing you the NIH clinical trial using mesenchymal stem cells for various diseases. And I guess the key for any clinical application is to overcome some of these obstacles in understanding more about the physiology and the property and the phenotype of the stem cells, how to use really a good methodology to expand these cells and study them in vitro, what are the signals that differentiate them into different specialized cells, or to keep them in their stemness status, and how to make an entire organ from a single stem cells, and how to improve the effectiveness of using these in a clinical trial. And when we overcome this, there is also more to come, is how can we make sure that when we inject these cells into the patient, that they home into the right site, they don't differentiate into a wrong subset of cells, they don't cause tumor, and how should we deliver them? To the right, directly to the site of, to the injured site, or systematically? And what cells do we use? Like, can we use mixed cells, or it has to be a pure cell population? And the source, like, do we use induced pluripotent, embryonic, bone marrow derived? And how to monitor their short and long-term therapeutic response in vivo and in vitro. At King Faisal, we have two components. Basically, we have the cord blood bank, where we provide a lot of storage for cord blood that can be transplanted directly into the patient. And we have a stem cell research that I will tell you about. At the cord blood bank, started in June 2006, and we have collected more than 4,300 units so far. This only represents 8 to 10% of our only hospital need, so we still need to do more. The target is to collect 1,200 at least units per year, and to have 15,000 units in total to have a center self-sufficiency. Out of these, we transplanted more than 117 units into patients that suffering from a different disease ranging from malignancy, immune deficiency, and hematological disorder. At the research center, the stem cell research is we basically focus on multiple aspects using the facilities that we have. We focus on studying the stem cell biology. We also have genomic and proteomic facility to study the different genes and the different proteins regulation during the different status of stem cell differentiation. We have a cell sorting facility and imaging facility that allow us to sort a single subpopulation based on 9 color staining. And we also privileged to have an animal facility that we can study the application of these cells in under physiological condition. And the goals of this is to identify a stem cell specific biomarkers, to be able to compare the stem cell from a different sources, and to compare also stem cells from normal as well as from tumor tissues, to identify the pathways that regulate the stem cell function and how do we target these by drug screening, and to study their physiology in vitro and of course in vivo in an animal model, as well as their clinical application. In the next few slides, I will focus on some of the studies that we conduct at our lab. And one of these studies is basically focused on targeting cancer stem cells. And the reason by doing so is that we found that most current remedy right now target differentiated cancer cells. Unfortunately, small population manage to survive, and these population, they have a stem cell-like feature, and therefore they are called cancer stem cells. They manage to regrow again, and they give a tumor relapse. So the hope is by identifying the different markers or the specific marker for cancer cells and the pathway, we can target these population and not only kill differentiated cancer cells, but also we kill cancer stem cells, and therefore we get a tumor destroy and hopefully eradication. To do so, what we look at is, this is again a busy slide, but we found that cancer stem cells and normal stem cells, they share a lot of similar features of the self-renewal ability, differentiation ability, long-lived resistance to chemotherapy, and their ability to survive in an adhesion-independent environment, and therefore they are able to metastasize. And we have recently identified a protein that regulates this process that acting through a gene known as NF-kappa pathway. And this protein is named FATN. This protein has been shown to be expressed by us and others under normal circumstances in mature dendritic cells, it's an immune cells, as well as in neurons. But it also been shown also by us and others that it's expressed in many transformed cells, including breast. So what we did is we focus in breast cancer because the advantage in breast cancer, we can get a normal samples, but you cannot get a normal samples for a brain or for eyes. So what we did is we bring breast cancer cells that express this protein, and we downregulate the expression of this protein using a short hairpin RNA to downregulate it at the protein level. And then we look at the different features of stem cells from the stem cell marker, self-renewal ability, and their resistance to chemotherapy. This is a histogram from a flow cytometry showing that the cells expressing the protein of interest, FASN, and this is after downregulation showing the cells lack the expression of protein. And what we found is that when these cells downregulated the expression of FASN, they lack the expression of one of the features or marks of stem cells, the ALDH. They also lack the expression of the cancer stem cell markers, which is the expression of high CD44 and low CD24 in a dot plot. This is, again, I'm sorry, this is a flow cytometry dot plot, each dot representing one single cell that express either CD24 or 44, which is two of the markers that's used to identify cancer stem cells in the breast. We found also when we downregulate FASN expression, we also downregulate the expression of the Notch gene, which is one of the genes important for the self-renewal pathway. Most importantly, when we take these cells that either express FASN or they don't, and we inject it into animals, they both grow. But when we treat these samples with doxorubicin, one of the conventional chemotherapy that's using in the clinic, we found that the FASN positive don't respond as much as the FASN negative cells. And as you can see here.
Dr. Mandir, time is...
How many minutes, sorry?
We're already 3 minutes beyond.
Okay. So, and we found also that these samples, they resist chemotherapy or patient who express FASN, they have a shorter survival than FASN negative patient. So the summary is that FASN expression in breast cancer cells regulate cancer stem cell features, and it's an indicator of chemoresistance. Unfortunately, I wouldn't be able to present these, but I'm going to go to my take-home message, which is I think stem cell therapy is coming, and it has a promising results. And I think there is several factors that control the stem cells function at a multi-step. And the better we understand, I think the better we can plan with minimal side effect. At the end, this is a teamwork. I would like to thank our team at the research center, as well as our collaborator from pathology, oncology, surgery, neuroscience, and the research center administration under his support, Dr. Sultan Steri. Thank you very much.
It is with pleasure that we present the recipient of His Royal Highness Prince Abdulaziz bin Ahmed bin Abdulaziz Prevention of Blindness Shield, Dr. Ting Zi. Dr. Ting Zi has a very long, as you would expect, CV and a very extensive one, but I thought I'll go through the main highlights of his research, and it is based on his presentations and his interest that we have structured this session around the stem cell research and its use in ophthalmology in particular. Dr. Zi took his bachelor's degree in agronomy in Beijing Cultural University, and then his master's in plant genetics and breeding, which was his main interest as he grew up in the rural part of China. He went for his PhD to continue his education in the same field, but it was in the US that he switched a little bit his interest, and his PhD was in molecular biology and biochemistry. He is currently, since 2008, an investigator working on germline stem cell regulation and germ cell development in Drosophila, which is the fruit fly, and investigations of molecular mechanisms controlling retinal stem cells and potential applications of retinal stem cells in the applications in the treatment of glaucoma and other retinal degenerative diseases. He, within a year of his stay in the US, as he has stated before, he realized that creating new crops wasn't for him. Although producing better vegetables or making better looking flowers is interesting and useful, he was more attracted to the mechanisms and how things happen, and this is how he moved into molecular and biochemistry research. He started thinking about stem cells and working on stem cell research when he moved to the Carnegie Institution of Washington to work with Dr. Alan Spradling on, as we said, Drosophila. And once he started the fly research, he decided to pursue the science as his own career. The main or the most important probably and the turning point of research that he had on stem cells was in 2000, where he discovered that stem cells divide into other stem cells when they are in contact with capsules around them. Otherwise, they would differentiate into the different types of cells. This was totally a new discovery that revolutionized the field, as it was previously believed that stem cells divide on their own without the influence of an external factor. It was when Dr. Robert Rich invited him to a glaucoma foundation function in the US that he started picking up interest into retinal cell and retinal stem cells and ocular diseases, and started his research focused on eye diseases. And in 2000, he moved to the newly established Stowers Institute and forged ahead with a string of new discoveries. These are some of the main discoveries that he made recently, where he pinpointed the chemical signals that the capsules use to tell the stem cells what to do. He identified the glue known as E-cadherin that tethers stem cells to the niche, preventing their loss. In addition, he also learned how to build the niche from scratch during early development. He's currently working on mice with the hope that they can regenerate or generate photoreceptors, retinal ganglion cells, and pigmented epithelial cells in the petri dish. This can then be transplanted into the mouse retinal degeneration models to test their potential in treating degenerative eye diseases, including glaucoma, retinitis pigmentosa, and macular degeneration. We have to prove, as he stated, that this works on mice first before this test can be then transferred and eventually tested on patients. He has a long list of professional affiliation. For the interest of time, I will not go through them, as well as academic awards. The last one was the one received this afternoon, and many publications in high reputable journals in science and in ophthalmology. As well, he's a member of 9 scientific advisory boards, including the research director of the Vision Research Center at the University of Missouri at Kansas City. Dr. Ting Tse.
First, we'd like to thank the organizing committee for inviting me. And I'm really happy to be here. It's a great honor to be here, especially for the warm weather. You can see here that I took a picture from my front yard. You see, even just 2 days ago. It's very cold. I'm very happy to be there in the warm weather and see some old friends, and also looking forward to make new friends here too. So I'm going to share with you today and what we can use stem cell to treating retinal degenerative diseases. As Professor Menzer already mentioned to you, different stem cell type can be used for treating degenerative diseases, namely embryonic stem cells, induced pluripotent stem cell, adult stem cells. I will not go into great details. Pluripotent embryonic stem cell can generate older cell derived from 3 germ layers, namely older cells in the adult body. They have advantage actually for use of embryonic stem cells that can be expanded indefinitely in vitro, have ability to generate various cell type for treating different disease, but it's still challenging to differentiate embryonic stem cell efficiently into particular cell type. You know, it's good they can generate many different cell type, actually very hard to direct them into particular cell lineage. They still have immunorejection problem. You cannot use can make your own embryonic stem cell, and teratoma problem, and also ethical concern. So the induced pluripotent stem cell come to the scene. That's why Yamanaka pioneered in 2006 and got a Nobel Prize 2012. And really the scientific world recognized actually the importance of discovery and potential application in treated degenerative diseases. Just like a ESL can be expanded indefinitely, and have ability to generate all the cell types in the adult body, and different cell type for patient-specific iPS cells like immunorejection. That's a huge advantage over embryonic stem cells. They still have, just like an embryonic stem cell, they're still challenging to differentiate them to particular cell type, still can form teratoma. Another type of stem cell, actually, they resist almost every tissue and body. They're equally actually useful, and in treating degenerative disease, it's adult stem cells. Here, actually showing you that, like a hematopoietic stem cell, they can generate all the cell in the blood system. Like here, I illustrated the retinal progenitor cell. They can generate all the cell type in the eye. But it's only limited potential. Actually, they can only differentiate cell that in tissue they reside. And it's relatively easy to guide them to differentiate particular cell type you want. And usually no tumor formation. For example, retinal progenitor cell we cultured. We transplant to many animals when they see tumor formation. Some of them have been tested in clinical applications. And disadvantage, as you know, the adult stem cell very hard to be cultured, expanded in vitro. Today we talk about how we can use the stem cell to treat age-related macular degeneration. As you know, the defective RPE cell causes loss of photoreceptor cells. And the retinitis pigmentosa, either defective RPE or photoreceptor themselves. Another is glaucoma, so due to loss of the retinal ganglion cells. So in order to treating this retinal degenerative disease, particularly for the photoreceptor degenerative disease, for AMD, we not only replenish photoreceptor cells, but also need to replace defective RPEs. For RP, either you replace the defective RPE only, and if the photoreceptor still there, or if the photoreceptor also lost, you also need to replace the lost photoreceptor as well. You can generate RPE cells either from human embryonic stem cell, or induce pluripotent iPS cells, or culture RPE cell from the cadaveric eyes. Human embryonic stem cell actually has been used to derive RPE cells, and actually this is pioneered by the Takashi from Japan. They use a stepwise differentiation protocol. Basically, you turn the embryonic stem cell into the eye field cells, then you can differentiate either to retinal progenitors and the RPE cells. And for human and mouse embryonic stem cells, they use different cocktail of the growth factors. And nevertheless, the procedure is very similar. I will not get into details. And many groups from all over the world have actually differentiated human embryonic stem cell into RPE cells. It's just here just shown that side of work from the Rubinov group from Israel. They successfully, efficiently differentiate the human embryonic stem cell into the RPE. They express during early culture time, they express MITF PAS6 markers. Later on, express mature RPE markers as the 01, RP65, bestrophin, and also have the pigment as well. More importantly, if you transplant this human ESL-derived RPE cell into the RP model, a rat model, they can protect photoreceptor from degeneration and also regain the light response compared with control, indicating the human embryonic stem cell-derived RPE cell are functional in vivo. And another approach actually inspired by recent work from the SASE group from the Riken University, in the Riken Center for Developmental Biology. Actually, you can grow a human ear cell and form entire retina, optic cup. And you see the optic cup, you have the RPE outside, you have retina inside. You can cut a piece of the RPE from the optic cup used for treatment. I think maybe it's a very good approach for produce RPE for transplantation. The human embryonic stem cell-derived RPEs are functioning in vitro, in vivo. Many available human embryonic stem cell lines. Disadvantages, as mentioned for embryonic stem cell immunorejection as an ethical concern, and still have a concern at teratoma formation as well. And that's why actually now many scientists get interested in the iPS cells. And many labs have tried to derive RP from iPS cells. Again, the Takahashi group and many other groups have successfully derived RP from iPS cells. And this here just signed one, showing some RP derived iPS derived RPE cells compared with the native human RPE. They express very similar molecular markers. And furthermore, they also show the apical basal polarity, and know the RPE has. This here shown that the sodium potassium dependent ATPs is localized at the apical site, and also can undergo phagocytosis. And so that's really, at least in vitro, the RPS-derived RPE cell appear to be functional. Further, that you can show that they have shown that these RPE cell, they have the functional voltage gated sodium ion channel. You can see this the native RPE. You can give apply the current, then you can see the depolarization, and then the iPS-derived RPE have similar properties. And another approach, as you mentioned, just like a human ear cell, you also can derive RPE from optical vesicles. That this done by the GAM group from the University of Wisconsin. They actually first culture human RPE cell and form optic vesicle, not really the optic cup, optic vesicle. That really express, CHEX10, really look like the retinal progenitor cells. If you expose the optic vesicle to the activin, they can efficiently turn into RPE cells and express pigment and really have RPE properties. I think human iPS-derived RPEs actually have a huge advantage over ES-derived RPEs. One is no immunorejection, no ethical concern, also can efficiently generate RPE just like ES cells. And one thing that concerns none of the iPS-derived RPE cell has directly been demonstrated to be functional in vivo, through the transplantation. I think based on the in vitro characterization, that probably is functional as well in vivo, but nevertheless needed to experimental verification. And the last approach you can generate the RP cell is you can culture RP cell from cadaveric eye. And the work from the Sally Temple group in New York has successfully cultured RPE from cadaveric eye, and then can reform functional RPEs in vitro. So I think actually this approach actually could be very good one. Very good one. So because we know that RPE is native and functional RPE cells. No ethical concern, easy access. You can get this, the RPE from iBank to expand them. And the disadvantage is still only few labs can expand these RPEs in vitro and still have the immunorejection problem because you can only get RPE from from cadaveric eye, not from the patient own. And next, we talk about how we can replace lost photoreceptor cells. And this approach has been inspired by the seminal work by Ali Lab in the University College London that first shown that you can transplant photoreceptor progenitor cell from P1 mice into adult mice. They can integrate and further prove they can form connection with the bipolar cells. Then they can actually have the functionality. They shown that they can restore light response of the rhodopsin mutant mouse retina. This tells us if we can generate photoreceptor precursor cell, you can use them to treat degenerative diseases. And you certainly can use the human embryonic stem cell, iPS cell, and the human retinal progenitor cells. As I mentioned earlier, you can use the human ES cell, use stabilized differentiation, and generate photoreceptor cells. Many groups from all over the world have accomplished this. Here I just show that the GAM group had popped in the PNS a few years ago. You can see this human ES-derived, actually the early retinal progenitor cell that express all the early retinal progenitor marker, OTX2, RX2, SOX1. These human embryonic stem cell divided retinal progenitor cells can differentiate the photoreceptor cells, which express known photoreceptor markers. Furthermore, if you transplant them into the supraretinal space of the mice, the mutant mice, the correct mice, actually no photoreceptor cell, they can regain light response, indicate that the human embryonic stem cell derived photoreceptor cells are truly functional in vivo. Another approach you can isolate the photoreceptor progenitor cells is you can use the human embryonic stem derived optic cups, and you can isolate retinal progenitor cell from the optic cup and generate photoreceptor cells. And also you can use the iPS cell to divide photoreceptor cell. As mentioned, iPS cell behave very like embryonic stem cells. And several groups have successfully convert human iPS cells into retinal progenitor cell and into the photoreceptor cells. Here shown that the human iPS cell also can form optic vesicles as well, express the marker for the retinal progenitor cells. They can differentiate into photoreceptor cells. Then actually you can use pegclamp that shown that have the normal function, have the property of function photoreceptor cells in vitro at least. And last project I mentioned, you can use human retinal progenitor cell instead of use the human embryonic stem cells. And several groups have cultured the human retinal stem cell from the fetal eyes and shown they can differentiate into photoreceptor cells. Because the fetal retinal progenitor cells still have the ethical concern because they're just like embryonic stem cells. So we decide, could we actually to identify the retinal source from adult retina? First, we use a mouse as a model, which is the eyesight, adult retinal stem cell from the adult mouse eye, and we expanded stem cell. I'm going to talk about it in my next talk. Then we label this the stem cell and differentiate into photoreceptor cell. You can transplant this in a retinal space, supraretinal space. You can see this photoreceptor can integrate, express the photoreceptor marker in vivo. This is actually the GFP labeled, fluorescent labeled, the retinal stem cell derived photoreceptor cell in the supraretinal space of the RD1 mice. As you know, RD1 mice is the mouse eye completely lack of photoreceptor cells. If you do the ERG, no light response. And very interestingly, after transplantation of the photoreceptor cells into the RD1 mutant mouse eye, and this eye regain light response, indicating that retinal stem cell, adult retinal stem cell can generate functional photoreceptor in vitro and can be functioning in vivo. So lastly, I will talk about what the potential, the general source for general retinal ganglion cell for treating glaucoma. Certainly, as I mentioned, human embryonic stem cell can generate all the cell type. Certainly can be generate retinal ganglion cells, and also iPS cells. And you can use retinal stem cell to derive retinal ganglion cells. And several groups have used human embryonic embryonic stem cell and iPS cell to generate actually the retinal ganglion cell in vitro. I should point out all this retinal ganglion cell generate from the embryonic stem cell, iPS cell, all mouse source. There are no human ESL, iPS cell derived retinal ganglion cells. I think this needed to be further investigated. Just here showing you the mouse iPS cell can generate actually the early retinal ganglion cell progenitor cell express MES5, PR3B, islet 1. They can generate mature retinal ganglion cell express the Psi 1.2. And so we also use a mouse retinal stem cell. We can generate actually very efficiently into, differentiate them very efficiently into retinal ganglion cells. Here we can up to almost 40% of cells become retinal ganglion cell. Actually never achieve such a high percentage use the embryonic stem cell iPS cells. If we transplant this retinal ganglion cell, and which also labeled with GLP, into normal retina, they never integrate. Very interestingly, if we injure retina with NMDA, the transplanted retinal ganglion cell can integrate and send axons as well. That's very exciting. But so far, we haven't observed the optic nerve reach, the axon reach optic nerve. So still a long way to go before we can use retinal ganglion cell to treat glaucoma. I think the major challenge for using stem cell derived retinal ganglion cell to treat glaucoma, difficult to generate large quantity pure retinal ganglion cell from any stem cell type for transplantation. That's one really major challenge. Still remain unknown if a transplanted retinal ganglion cell can make correct synaptic connection with remaining retinal neurons. That's not known. Although we see the integration, but we don't know that make the function connection. It remains unknown whether transplanted retinal ganglion cell can grow long axon to reach and correctly talk to the LGN in the brain. The conclusion is the human embryonic stem cell, iPS cells, adult stem cell, fetal progenitor cell could be used to produce desired retinal cell types and RPE for transplantation. Different stem cells and approaches have their own advantages and shortcomings. Should be tested experimentally on animal models. I should point out that many people interested in using stem cell to treat retinal degenerative disease. And the field really need large animal model, retinal disease models for testing stem cell functions. So I should point out if some research here interested in develop large animal disease, retinal disease model will be important for the stem cell application in treating eye disease. In the last, I'm going to note that people from my lab have contributed to the retinal stem cell research. Thank you very much for your attention.
Thank you. The next presentation for Dr. Mohammed Al-Jum'a, Stem Cell Research at King Abdullah International Medical Research Center. Dr. Al-Jum'a.
Assalamu alaikum. I'd like to thank the organizer for giving me this chance to share with you a small introduction about King Abdullah International Medical Research Center, because I'm sure that some of you haven't heard of the center, as it's just established or under establishment still. And then I'll share with you some of the research activities that are done in the center. King Abdullah International Medical Research Center is considered to be the research platform for the National Guard Health Affairs, as well as King Saud bin Abdulaziz University for Health Sciences. It was established in 2007 with a 10 years planned to be fully operational in 2017. Currently, now we're under the establishment phase, and that phase has really gone multiple steps in building the more important aspect and establishing multiple initiatives that will lead to that extension. I'm sorry that it went into the vision of the center is to be a research platform for better health, and the model that they are using is a full translational research model. Before really going into the research activities, we felt what would differentiate us as a research center and a research institute in Saudi Arabia and in the region really to go on and build the strategy. And the focus was built on really having the integrated full academic as well as an extended state-of-the-art health research facilities, and those would differentiate us from any other research institute. In addition, the presence of really multiple program that is not really to be focused for only a tertiary health research center. We are a center that provides primary, tertiary, and care, and that would make us a very good platform for population research. We have invested a lot in the ethics, and the main thing is really to keep the trust between us and our population to continue the research activities. With those in mind, what we have built is really to build our strategic goals, and that focus really in building the human capital, organizational efficiency, as well as the business growth to capitalize on our main work, which is the research activities, which will be both basic research and population-based research. The goals goes into multiple initiatives and multiple platforms. One of those is the cell therapy program, and this is where I'm taking you into what we are doing with the stem cell research currently. And we have been focusing on cord blood, but we have took different initiatives, and that initiative is really going into the model of where we should be really focusing our activities. And the focus is going into both cord blood research, and our focus not in only treatment, but in really improving the quality of extraction as well as the immune response to it. And the second one, rather than really going into the fetal or the adult stem cell, we focus our research on the placental stem cell. And we have been focusing into both collection and extraction, as well as characterization and doing cell therapy on those. I don't really know to go into the introduction about the stem cell because the previous two speakers have done a very good job on that. But I'm really going to focus why we use the placental stem cell. And if you see the placenta, it has some fetal component on it. And what we did over the last 3 years is really trying to extract stem cell from the placenta, and we focus on extracting the fetal component of the placenta. And to initiate this work, we have really worked into using boy fetuses to make sure that when we extract the stem cell, it has Y chromosome, and this would give us the most component of mesenchymal stem cell that have a lot of properties of fetal component. We have worked a lot in characterizing these stem cells and growing them, and the work in really getting the way of getting the cells out and culturing them. We pioneered a new way, and it has been published. As well, we have went a long way on characterizing those fetal stem cells and looking into their male characteristics. Again, that work has been published. Why we focus on the placenta? Because the placenta has a lot of stem cell on it. It's easy to extract. It's usually discarded, and that we felt that really, rather than as a research center, rather than focusing on what other research facilities in the kingdom are focusing on, we went into the placental stem cell. What I'm going to work on, actually, in really showing you, as well, we didn't really focus on regeneration. We have worked a lot in really using the immune properties of the stem cell and using that into therapeutics. And what I'm going to present to you is only one of these studies that most of the published work on the stem cell has been really focused into the immune properties of the dendritic cells. We felt that there's a lot of work has been done on this. What we have focused on is into the macrophages. And I'll introduce you if you know the macrophages, there is the M1, which is the pro-inflammatory macrophages, and those are implicated in the inflammatory disease in general, as well as rejection, and stimulating any inflammatory response with either for transplant or for other autoimmune diseases. And the M2 is the other macrophages which work as immunosuppressive cells. And they promote the T helper cells to downregulate the immune response. The study objectives was to see if we can modulate the function of the monocytes by using placental stem cell. This work has been done on some of the dendritic, but not on the monocyte stem cell. And then we went into see the differentiation, whether it goes to either M1 or M2. As a method, what we did is we took the placental stem cell from the chronic villi, and using the trypsin, we were able to extract the stem cell, and those stem cells were, this is a picture really of showing the spindle stem cells that we were able to extract from within 14 days from the villi. And then we used a human healthy donor, and we used their monocytes. And then after that, we have really intubated the monocytes with the mesenchymal stem cell and look to what will be the responses. We have used a culture which used transwell chambers with small bore holes, which really allow, and we did it in 4 ways, either to immediate contact or with all using the secretome rather than reusing the contact of the cells. We did the analysis flow cytometry as well as ELISA test, and our results showed that the co-culture of placental mesenchymal stem cell with monocytes stimulated the inflammatory M1 macrophages to differentiate into M2-like macrophages, which is really, we have modified the differentiation into much more immune modulator rather than pro-inflammatory responses. We have looked into this into either looking into the morphology of the cells, and I'll show you a slide looking into the morphology. As well, we looked into all the surface markers of the stem cell, of the M2 macrophages and compared them to M1 macrophages. As well, we looked into the function of these differentiated M2 macrophages and whether they can reduce the inflammatory response. And we have used the marker of CD40, CD80, and CD86, and those did show very well to show that whatever differentiation was induced by the placental mesenchymal stem cell is really, as well as morphological, is cell functional. As well, we looked into the other marker with which will show the function as well. This is the typical M1-like macrophages, which is usually egg-like cells, and when we intubated them, they change into the spindle cell, which is the M2 macrophages. Just to summarize the whole process that was done, we took the monocytes from blood, and now we are going into much more monocytes with a disease model, and with intubating them with a stem cell, either direct contact or through the secretome, which includes some of the generatives. And we have looked into the mechanism that they have induced the M1 macrophages, which is through the GI receptors. We were able to stop the differentiation of M1 and go more into M2, which is immunomodulator and usually help to do the suppression. I tried to went fast for the time's sake, but in conclusion, our finding is suggestive that the new immunosuppressive properties of the placental mesenchymal stem cells that may be employed in the resolution resolution of the inflammatory associated with inflammatory diseases or tissue repair, and this is really a way of looking into it. To carry this further, we have just got funded for another project which would look into using the placental either direct contact or the secretomes into an animal model for the experimental immune encephalomyelitis, and hopefully that would show that the effect is not only in vivo, but it will go in vitro. I'd like to thank you again for allowing me to share with you the introduction about the center and some of the basic work what they are doing as we are in the initial stages, and thank the contributors, and most of the work has been led by Dr. Mohammed Abu Mar'i. Thank you.
Thank you. Thanks a lot. The next two presentations will be by Dr. Ting Tse. The first of which is application of stem cell niche concept in the field of ophthalmology, then derivation of functional retinal cell from the adult retina. Dr. Tse.
So the next I'm going to share with you our recent work derived the function stem cell from the adult mouse retina, and we want to use as a model to investigate the possibility use adult stem cell to treat it in treating retinal degenerative diseases. As some of you may have already known, that people thought have stem cell in the cellular body can generate different retinal cell type. And they published series nice paper, but recently has been disputed. And we also be able to generate as publication. We have shown that actually the cellular body also can generate neurosphere, really consistent with the recent finding. These cells could not efficiently generate any retinal cells. They're still like RP-like cells. Then also some of you already know that a mural cell, in the only glial cell in the retina, also been proposed function stem cell in the retina, has been thought they have ability to repair injured retina. And they also have been cultured in vitro and used this culture cell to generate the different retinal cell types. We also culture these cells. We find it's very hard to generate any, actually efficiency is extremely low. It's not very useful. We decide to take a naive approach. Let's see whether we can derive really the good stem cell line from adult mouse retina. And we actually isolate the clean neuroretina and dissociate and culture in vitro. This takes more than 2 years hard work and lots the financial investment in order to figure out the best condition. We will be able to finally we'll be able to culture a populated stem cell and express SOC2 nesting. And there's a marker for neural stem cell. And the eye belongs to the central nervous system as well. We expect that the stem cell should express these markers. Furthermore, they also express the PAS6. More importantly, if they put this stem cell line in the differentiation condition, basically withdraw the cell renewal growth factors, they can spontaneously differentiate into retinal ganglion cells, macular cells, horizontal, bipolar, or photoreceptor cell. All the 5 major retinal cell types. Also, the glial cell, the Mueller cells. Actually, upon that, you can look at the picture very efficient if you suppose, can very efficiently differentiate this cell into retinal cell type, in contrast to the cultured Mueller cells and the retinal stem cell from cellular body. I think that we, as a basic stem cell biologist, we are very excited about the cultured stem cell line. We actually want to use to study regulatory mechanisms. And we also want to use this cell line to start the lineage specification. I mentioned in my last lecture, is one of the major tasks in use stem cell treated disease is to direct stem cell efficiently into particular cell lineage. That is the main challenge for using stem cell in the future regenerative medicine. We can use this cell line. We find, for example, the FGF facilitate the differentiation of the retinal stem cell into retinal ganglion cell. You can increase from like 10% almost to the 30%. So it's very exciting to us. And furthermore, you can use this stem cell also differentiate to other cell line, may be able to generate all the cell, any cell type you're interested in. One major reason for us to derive this stem cell from retina is treated in retinal degenerative diseases. And we want to test whether it could be used to treat glaucoma. And first, we figure out that one very good condition efficiently differentiate actually the stem cell into retinal ganglion cell, up to almost 40%, just mentioned. And if we label this retinal ganglion cell with GLP, just I mentioned in last lecture, I will explain to you a little more. If we transplant it into the vitreous of the eye, into normal retina, this cell cannot integrate. They only cumulus surface that some of the still express the marker of the retinal ganglion cells. If you use NMDA, the drug, to injure the retina, actually get rid of some endogenous retinal ganglion cells, the transplanted retinal ganglion cell can integrate, send axons. And as mentioned, but this transplant retinal ganglion cell could not send axon to optic nerve to the brain. So the major challenge is retinal ganglion cell, retinal stem-derived ganglion cell treated glaucoma. It will prove retinal ganglion differentiation efficiency. The efficiency is still low, 40% is still low. You want to use the therapy, you probably want to get a pure, close to pure, the retinal ganglion cell population. And also improve integration efficiency. I just assume that some can integrate to the many transplanted cell, still could not integrate. So we need to still figure out how to induce. And furthermore, we need to figure out how to correctly target the axon of these retinal ganglion cells into LGN. In the early development, they have the cues extract the axon to LGN, but this cue disappeared in the adult retina. That's what we need to figure out. And then lastly, we need to figure out how make the retinal ganglion cell to form right connection and integrate into correct neural circuitry in the retina. I think in order to use retinal ganglion cell to treat glaucoma, it's still a long way to go. As I hope will convince you, maybe retinal ganglion, the retinal stem cell could actually used to treat the photoreceptor degenerative diseases. We also developed the culture condition can differentiate about 50% retinal stem cell into photoreceptor cell. You may not, this is not 100%. I think it's the highest that we know, differentiate, any stem cell into photoreceptor cells. If we label this photoreceptor with GFP, you transfer supraretinal space, they can integrate into the retina and also express GFP as well. More importantly, you look at these two integrate the photoreceptor cell, have a normal morphology, have R segments, have a long axon to the outer plexum layer where they can form synapse with bipolar cells. So that's very exciting. They not only also integrate, can form synapse. Further, we've shown that if you use this GFP labeled the photoreceptor cell in the RT1 mutant mice, which lack a photoreceptor cell, you can restore light response. This really makes us very, very excited. That means that this adult stem cell could be used to treat retinal degenerative disease, particularly photoreceptor degenerative disease. And so this finding raises the possibility really culture human retinal stem cell might be useful to treat photoreceptor degenerative disease. This work just accepted publication in one of the Nature journals called Cell Research. It should come out soon. Hopefully, if you're interested, you can read it. We actually also try culture human retinal stem cell. We'll be able to culture the human retinal stem cell, express the stem cell markers. And you also can differentiate to the, very efficient to differentiate to the mural cells, right? It's just like the muscle retinal stem cells. We also can differentiate to the photoreceptor cells as well. And we still need to test whether human retinal stem cell divided photoreceptor cells are functional in vivo. We are doing right now. We still don't have answer yet. And even so, the cultured human retinal stem cell could be used to treat retinal degenerative disease. That still have one more challenge: is immunorejection. Could we generate patient-specific, retinal cells? Instead, use the ES cells in iPS cells. As you know, they can form teratoma. We decide to avoid potential immunorejection and also the teratoma formation. We are working on reprogramming fibroblasts directly into retinal progenitor cells. Recently, we'll be able to convert fibroblasts into neural stem cell-like cell that can generate functioning neurons in vitro. This is very exciting. We are working on right now, turn the fibroblast to induce the retinal progenitor cells. Hopefully, this retinal progenitor cell can be used to produce photoreceptor cells, RPE cells, or retinal ganglion cell. So we are working very hard on this. Hopefully, in the near future, you will hear good news on this approach as well. So I'm going to conclude my talk, acknowledge the people, this group of people again, for their contribution to the retinal stem cell research in my lab. So we're going to switch to another talk. It's about, this talk not about actually exact data, just show how apply some stem cell concept into field ophthalmology. A special niche and why the niche is important, I'm going to explain to you. As you know, the stem cell, as I introduced earlier, can be very useful. We also think about it is the embryonic stem cell, and just already mentioned, adult stem cell equally important. And adult stem cell not only maintain tissue homeostasis, they also involve aging, the cancer as well. I think it's really important to understand the regulation of adult stem cell in vivo. Actually, the adult stem cell exists all different kind of creatures, from human to the fruit fly and the worm. And actually, the most important stem cell concept come from studies of the model organism such as fruit flies. That's my lab has in last decades has used the fruit fly stem cell as a model to investigate how stem cell is maintained in vivo. And we've shown that the stem cell is maintained by the environment. So how we show that? That's I know you're a medical doctor. I can show you on one slide how we demonstrate, elegantly demonstrate stem cell regulated by environment. Here, showing you this stem cell, red cell stem cell, is a differentiated daughter. See, this red cell always divided as stem cell, a daughter always move away. We don't, when the popular idea is, well, these two stem cells are born differently. That's why one is a stem cell, one is a differentiated daughter. We find if we put the usually differentiated daughter in the same environment, actually both become stem cells. That means that two stem daughters are not different. If we live in the same environment, both can become stem cell. This is a direct demonstration. Actually, stem cell is determined by the environment, not by themselves. So the leading to widely accepted called stem cell niche. Actually, the group of cells in the tip we call cap cell as a niche cells to regulate, control stem cell identity. Why that's important? The niche is the number one principle is several new niche exhibit structural symmetry. So here actually take from a recent review showing that this is the different stem cell niche. This is the C. elegans worm. You see it's one cell niche is niche, this stem cell. This is just fly over, I just mentioned, this niche is stem cell. This is the fruit fly, the testes. This is red cell stem cell, the green cell niche. This is the mammalian hematopoietic stem cell and neural stem cell. You can see stem cell always look to one side, niche on one other side. The differentiated cell move away from the niche. In a way, actually stem cell, the niche exhibit a structural asymmetry. Why this important? To allow the stem cell and their differentiated daughters to interact with different surrounding cells and to be regulated differently by distinct signals combination from the surroundings. This also allowed antagonizing several new differentiation processes proceed simultaneously in the same tissue because several new and differentiation is the opposite, is antagonizing process. Because the symmetry allows the two processes to be coexist in same tissue. How we can use this concept in the field of the ophthalmology, especially we want the generate tissues, in future engineer lens, retina, and cornea. It is important to produce the biodegradable scaffolds in which one part actually promotes cell renewal and the other part stimulates differentiation. That way, you can generate a functional tissue in vitro. That's what the importance of the symmetry of the niche. Second principle, cell renewal niche produce short-range signal, maintain cell renewal by process differentiation. Actually, we shown in the elegant in the fly system and Drosophila system. This is the niche cell produce one BMP called BMP 2 and 4, and they directly activate BMP in the stem cell. Actually, not much activity in the differentiated cells. And very interestingly, when a differentiated gene known is a mass different gene, it's called BAM. Actually, the expression differentiated cell is repressed in the white type stem cell. In the BMP mutants, these different genes are upregulated. That means you need a niche signal to shut off a differentiated program, maintain cell renewal. If you have a lot of BMPs, now you have you have 1,000 stem cells instead of only 2 stem cells in the structure. So that's the niche factors very important for expand stem cell in vitro. That's why very difficult to expand adult stem cell in vitro because we know little about the regulation in vivo. I think the basic research is still very important for stem cell application. So we know the stem cells are controlled by the BMP signal and to maintain cell renewal by prevent differentiation. This is not only true for fly, it's true for human hematopoietic stem cell niche, also intestine stem cell. They also use BMP, they also use WIN, they use many other molecules, single molecule to maintain cell renewal. So I just want to point out that different niches use different combination of growth factor to maintain cell renewal. When you culture stem cell, your favorite stem cell, you don't want to use the condition other people use. You have to figure it out on your own. That's when the task you have to perform in order to expand stem cell in vitro. What is really a potential application niche signal in the ophthalmology? Well, in order to expand the eye-specific stem cell in culture such as lens stem cell, corneal stem cell, retinal stem cell, we must identify all the key niche signals for the controlling cell renewal. Even though the signals, you can expand them easily in vitro. Okay. Luckily, stem cell in the lens, the corneal, or retina could use different growth factor control cell renewal. I think that will be the case. That's why you have to really look at the different tissue, different stem cell type, try to come up with different conditions for culture. And the cell renewal niche uses adhesion molecule to anchor stem cell long-term cell renewal. Stem cells not simply stay one place for long term. Actually, they're moving around. Actually, most time in the adult tissue, stem cells are stationary because they are anchored in the niche through adhesion. We have shown that E-cadherin, the adhesion molecule, important to keep the stem cell in the niche in the fly ovary. And this actually, the anchorage is very important. If you remove the cadherin, move adhesion molecule, stem cell can leave the niche and differentiate and last. And as pointed out in the many stem cell system, they not only use the cadherin, the typical cadherin here, they also use the integrin, they also use the delta notch, or use many other the receptors to keep stem cell in the niche for long-term cell renewal. This adhesion not only for keep stem cell in niche, also help stem cell survival, proliferation, and also fight against aging in the normal tissues. You need adhesion molecule in stem cell niche. How we can use this idea in ophthalmology, especially in culture, your favorite stem cell, you probably consider use some, modulate your extracellular matrix, because we know integrin is very important for many adult stem cells. I think that's why we coat our plates with the different extracellular matrix proteins. In the tissue, in the culture, in the tissue engineering, and the biomaterial scaffold should have the adhesive properties for keep stem cell in place. Then the way you will not mix with the differentiated cell, interfere with the differentiation. Number 4 principle, I think people always try to use the different stem cell, use one stem cell from this tissue and to treat these other tissues. The most time it will not work. Why? I can explain to you. Stem cell is maintained by the intrinsic factor in combined with the niche signal. There's this image, just like a lock and the key. They need to work together to achieve the cell renewal process. We have shown that. And the niche signaling and the intrinsic work together regulate niche anchorage, also prevent differentiation for long-term cell renewal. So actually we use this idea, that's why I mentioned how we reprogram fibroblasts to the neural stem cell. We thought, well, you have some right intrinsic factor, have actually right growth factor. Indeed, we can use lineage-specific transcription factor, combined growth factor, can convert fibroblasts to neuroprogenitor cell, computer functional neuroprogenitor cells. So we believe you can figure it out in important transcription factor retinal stem cell on a growth factor for maintaining cell renewal. We can convert a differentiated cell back to retinal progenitor cells. So that's no magic. You need to really understand this cell, and we can make this cell grow in vitro. After we identified the critical niche signal and intrinsic factor, and we can convert actually any cell back into cell we desired. Principle number 5, with stem cell differentiation require differentiated niche. So this is a new concept which is published last year, 2 years ago now, not last year, 2 years ago. In the past, we thought, you have cell renewal niche, stem cell renewal niche, cell renewal. They leave the niche, they could differentiate. Turns out not the case. They need continued requirement for their own niche for differentiation. Here, we also use a flyover as a demonstration. This cell, this differentiated stem cell progeny, already leave the niche stem cell niche here. Now they're surrounded by this group cell we call differentiation niche. You would disrupt this, actually, this cell's function, actually associated with the differentiated cells. They also this stem cell cannot progeny cannot differentiate. So I believe this is what happened on the cancer stem cell formation. Actually, you mess up a differentiation environment, and some stem cells probably persist, then you can for tumor formation. So we believe actually stem cell actually progeny differentiation requires special marker environment, just like a cell renew, stem cell renew. So this is our current actually model for understanding that two different niches work together to regulate cell renew and differentiation. To engineer eye tissue, a general eye specific cell, don't forget actually build two niches. One is cell renew for stem cell expansion, one for differentiation, produce cells you want. Okay, that's really what we learned with the fruit flies, the basic research. In the last, I want to acknowledge the people who work on my lab in the last 10 years, contributed to the understanding stem cells, cell renew, and the niche function. Thank you very much.
Thank you very much, Professor Z. This last lecture or presentation concludes our session for today. Thank you for staying until the end. We appreciate your attendance, and I'd like to remind you that we have another session. It was supposed to start at 4 o'clock. Unfortunately, we're already behind schedule, 10 minutes behind schedule. So we will take a break of 20 minutes, and it's almost 10 past 4. So we'll take 20 minutes break, and we'll start at 4:30. So 4:30 we'll start the second session of prevention of blindness. Thank you.